Why? Humans have lost the use of their body. Now here's comes the science. Just relax a little bit. We just lost our ability to treat methanol like the animals treat methanol. It's real important for you to know the science behind it, just a little bit. And I try to make it as simple as possible. But basically, here's a cell, an animal cell, and the only things I'm going to ask you to sort of think about or learn about are just a few little parts of that cell. The cell, is full of a liquid. And it's very much like egg white. It has that consistency, and it's called cytosol. So the liquid in a cell is called cytosol, beautiful fluid material. There are things that float around in that cytosol, the two most important of which is one, the mitochondria, which we'll talk about later on, which is the powerhouse of the cell that makes all your energy. You people with MS, one of the big symptoms of MS is loss of energy. Where does that come from? Not from demyelination. That wouldn't cause loss of energy. We'll show you in a minute. And the peroxisome. The peroxisome is what makes us different than animals. Really important. This is extremely important. It's a marvelous structure. In my book, I try to size things for you and give you size relationships and this sort of thing. It's a pretty big structure. But what that is, there's 250 of these things in every single one of your cells, about 100 to 250. These are the detoxifying part of the cell. These are the things that take poisons and break them down into safe stuff. Okay? When methanol breaks down, the first thing it turns into is formaldehyde. If that happens inside this peroxisome, which is where the animal catalase is, which does that beautiful job of turning methanol into formic acid, which is extremely safe. It's a food acid. If that happens and you're safe, it's okay. It's like, it even looks like one of these bomb control things. Okay. Methanol goes in, formaldehyde never comes out. Only formic acid comes out. Formic acid is a food acid. The scientific literature doesn't want you to know that. They try to tell you that formic acid is what causes methanol poisoning effect. It's just nonsense. And the literature for methanol has been poisoned by Donald Rumsfeld and his crowd. They bought off a bunch of research. Anyway, I'm not going to go there. But anyway, the medical literature is laughable when it comes to what causes methanol poisoning. It's formaldehyde. Anyway, so this is the structure. It's just like this thing. I mean, these guys have got a hand grenade, and they want to blow it off safely, so they just throw it into the sphere that contains the explosion. That's just exactly what the peroxisome does. Here it is again. There's a cytosol, peroxisome, mitochondria. Our peroxisome, or one of the enzymes in it, has been mutated. This happened to us. I talk in the book about Adam and Eve kind of thing. It happened really early on. Every single human, their catalase enzyme and the peroxisome cannot metabolize methanol at all. It bounces off. So what does methanol do? Where does it choose to turn into formaldehyde? In the cytosol of the cell. There's this other enzyme called alcohol dehydrogenase, which is what we use to turn ethanol into vinegar, basically, in our bodies and make it safe. But it floats free in the cytosol. It floats free, and when it produces formaldehyde, that formaldehyde is let loose. It can go into the mitochondria. It can go into the DNA, and the DNA it methylates. Methyl alcohol makes formaldehyde. Formaldehyde methylates. You're seeing more and more where methylate causes diseases like autism. The major cause of autism is methylation. My lecture on Sunday, the 6th, will be concern itself with other diseases besides multiple sclerosis, and it'll be the Men's Working Club. Again, free lecture, just come and I'll tell you about what happens there. But anyway, so that's what makes us different. That's a huge difference. So we can't be equated with animals. This is what this beautiful alcohol dehydrogenase looks like. It takes ethanol and basically turns it into acetic acid, which gets converted to carbon dioxide. We get energy out of it. What does it do with methanol? Methanol, formaldehyde. And that's as far as it gets, ladies and gentlemen, unless you've had a massive dose of methanol. It goes to formaldehyde, and the formaldehyde attaches to your protein. It looks for a basic protein. The best basic protein close to where this happens is myelin basic protein, and it attaches to it and disappears. Only recently have they been able to detect it, and I'll tell you what happens at the end of this series when they did look for this, and they can look for it now. So what happens then? So what's wrong with that? What's wrong with that is that the innate immune system, which the drug companies have all targeted with their stupid drugs, interferon, I can't name them all, that is supposed to sort of stop MS or slow it down. None of them work really much better than others. They don't work at all. They're more like placebos. But they're targeting the white blood cell. They call it the leukocyte, the macrophage. These cells have sites specifically to identify formaldehyde modified protein. They're killer cells. No, they're not. They don't call them killer cells, but they're cells that are made to eat things that they have sites on them for, and they go looking for a myelin basic protein that's been modified, and they eat it. So when your doctor looks at, through a microscope at your myelin plaque, okay, you see desolation to the outside of the plaque, and what does he see? He sees a bunch of macrophages, or they call them leukocytes, devouring your myelin. There's nothing wrong with those leukocytes. There's nothing wrong with them. They're doing what they're made to do. Devour formaldehyde modified protein. Because formaldehyde modified protein does no good. It's damaged, it must be destroyed. And the innate immune system was probably developed first because of that. Because the body does use formaldehyde to do other things. And it can't risk losing proteins. I try to make this simple in the book. I use birds to exemplify these molecules. I'm using a pigeon for methyl alcohol. The reason I did that is because a pigeon was a certain size, and I tried to make some model so you'd understand how cells work. So if a methyl molecule is the size of a pigeon, then your nerve cell would be the size of Manhattan Island. Okay? And I try to give size and relationship to this kind of thing. So I use pigeons as for methanol, and I use these hawks for formaldehyde. So what happens is the alkyl dehydrogenase has a site on it where methanol can fly in, and it flies out as formaldehyde. But what most people don't know, including definitely all physicians that I've ever met, and a lot of chemists, is that formaldehyde has two distinct personalities. And this is the key. If scientists understood this better, they would understand where I'm coming from. There's the basic formaldehyde, which looks like this, which is a terrible molecule. It's very reactive. But when you dump that in water, when you add it to water, you produce the hydrated form, okay, formaldehyde hydrate, which is a monster. Okay. This is how it's depicted. It has a thing called resonance going on. It has two sites that have to grab on to something, and its protein is its food, basically. It's scary, isn't it? You're Kiwis, you don't get scared. An interesting thing that this causes, which again a lot of medical people don't understand, and that's an ability for formaldehyde to cause bridges, to link two proteins. Okay. Linking proteins is another way to cause the macrophage, especially if you're using formaldehyde, to get a macrophage excited. It activates the macrophage to see proteins that are linked together. That's something very wrong with that. And formaldehyde can do that only because it has these two personas, a basic and an acid persona. And this is the acid persona grabbing a hold of the rear end of the basic persona, and so on, and you produce these long chains. That's why formaldehyde in water will eventually solidify in the bottom. It's so reactive, it combines with itself. Very unusual chemically. But here is something that's not unusual. This is really interesting. It attacks protein, and this came out of an article that did show successfully, that extremely small amounts of formaldehyde can produce the tangles, the binding of proteins found in the brain into tangles, and these tangles are what they find commonly, well, they always find in Alzheimer's. So here we have, and this is not my diagram, this is his diagram. Here's a tau protein, which is the protein involved. It's a basic protein. There's formaldehyde. You get some attachment. Then the hawk and the crazy heart start binding to each other, and then you start binding them together. And he claimed that this is the only way that he believed the tangles can form. But he said the only problem is we can't figure out how the formaldehyde can get into the brain. And formaldehyde can't get into the brain. Seldom gets into the brain unless it's huge concentrations because of the blood-brain barrier. It's so reactive, if you breathe it in, it gets into your bloodstream, it disappears. It attaches to proteins along the way. Gets to the blood-brain barrier, which is a narrow gap between the epithelial cells and the lining. I'll show you a picture instead of trying to tell you these fancy names. Can't get to. How do you get formaldehyde inside the brain? To cause autism? Yeah, autism too. But Alzheimer's and multiple sclerosis? You can do it because the only place that you have alcohol dehydrogenation in your brain is in the lining